Hello everyone. Today we will know about the endomembrane system. So what is the endomembrane system? So endo means inside and the membrane means of course the membrane. So endomembrane system, that means the system that is present inside the membrane. So of course membrane means it is the plasma membrane. That means a system is present inside the plasma membrane is called as the endomembrane system. But before going to the endomembrane system, we should know something about the cell. So this is the cell and the outer layer is called as the plasma membrane. And inside the cell, of course, the cytosol, the white part, are called as the cytosol, or the liquid part or the watery part is called as the cytosol, and many organelles are present. For example, this is the nucleus. This is the nucleus, and inside the nucleus a round structure like this is present, and this is called as the nucleolus, and this is called as the mitochondria, and this is called as the peroxisome, and this is the ribosome that is present in the endoplasmic reticulum, and this is the vacuole, and this is the lysosome, this is the Golgi complex. So the cell organelles can be two types. One is called as the single membrane bound organelle, single membrane bound organelle, and another is called as the double membrane bound organelle. So what is the single membrane bound organelle? What is the double membrane bound organelle? For example, if we look the vacuoles, so here the vacuoles, here the membrane is one. That means this organelle or the vacuole is surrounded by a single membrane or single layer, that is called as the single membrane bound organelle. For example, if we look the mitochondria looks like this. This is a one layer, and inside that layer this structure is like this. So that is called as the cristae. So that means in the mitochondria there are two types of two membranes are present, and this is called as the outer membrane and this is called as the inner membrane. That means all the enzymes, all the materials that are required for the mitochondria, that is present inside the cristae or inside the inner membrane. So that means the mitochondria is a double membrane bound organelle. So that means the mitochondria has two types of two membrane. One is called as the outer membrane and another is called as the inner membrane. For example, the nucleus. Look at the nucleus structure. Here it has a two layer. This is the one layer, and inside it another layer is present. So the outer layer, of course, it is called as the outer membrane, and the inner membrane is called as the inner layer or inner membrane. So that's why the mitochondria, nucleus, and another is the chloroplast. Chloroplast. So it is also a double membrane bound organelle, because the mitochondria, nucleus, and the chloroplast, they have two membrane. That means the boundary has two. They have two boundaries. That's why it is called as a double membrane bound organelle. So all other organelles are called as the single membrane bound organelle. For example, ER or the endoplasmic reticulum, the Golgi body, lysosome, a vacuole, and all other organelles except the mitochondria, nucleus, and the chloroplast, they all are the single membrane bound organelle. So now come to what is the endomembrane system. So endo means inside, the membrane means the membrane. That means a system is present inside the membrane, that is called as the endomembrane. So what is the system? So that means a system is present and or it is responsible for the formation or production, modification, packaging and transport. So that means a system is present inside the membrane which are responsible for the formation, modification, packaging, transport of protein and lipid. So that means it is a system. That means a system is coordinated to form or to formation, modification, packaging, transport of protein and lipid. So what are the system? The system includes endoplasmic reticulum, Golgi body or Golgi complex or Golgi apparatus, and lysosome and vacuoles. So that means these four organelles are responsible for the formation, modification, packaging and transport of protein and the lipid. Look at the structure. This is the ER. This structure is called as the ER. So ER, after that it is the Golgi complex, then it is the lysosome, and then it is the vacuole. So that means these four organelles are responsible for the protein and the lipid formation, modification, packaging and transport. That's why the endomembrane system includes these four organelles. Why these organelles? Because these four organelles are cordially managed to formation of this protein and the lipid and transport, of course. And other organelles, for example, mitochondria, chloroplast and peroxisome. So they are also present, but they have different function. That means they have their own function. For example, this mitochondria, they have it have its own function. So that means they don't include in the endomembrane system. So endomembrane system includes the endoplasmic reticulum, Golgi, lysosome and the vacuoles. So if we look this ER and the Golgi, if we zoom it, it looks like this. So that means if I zoom it, it looks like this type of the structure. So now come to the endoplasmic reticulum. Why it is called as the endoplasmic reticulum? Endoplasmic reticulum. So endo means inside, plasmic means cytoplasm. So reticulum means it's a network. So that means inside the cytoplasm a network like structure is present, and that is called as the endoplasmic reticulum. Look at the structure of the endoplasmic reticulum. It is like a network like structure. So that's why it is called as the endoplasmic reticulum, because it is present inside the cytoplasm. So the endoplasmic reticulum, it was first seen in the electron microscope by the Garnier. But if in the exam, in the NEET exam, it will be asked that who coined the term ER, and the answer will be Porter. The Porter coined the term ER, or the endoplasmic reticulum. So the endoplasmic reticulum, it is found in all the cell except mature RBC and embryonic cell and prokaryotic cell. So that means, of course, the prokaryotic cell, they don't have the ER, and the eukaryotic cell, The mature RBC and the embryonic cell, they also don't have the endoplasmic reticulum. So now come to the structure of the ER. If we look the structure of the ER, the structure look like, there are two types of the structure. One is called as the cisternae and another is called as the tubule. So what is the cisternae, what is the tubule? Look at this structure. This flattened like structure is called as the cisternae. So they are the cisternae. So what is the cisternae? The cisternae is the long flattened unit. Long flattened unit, and they are the hollow. So hollow means it looks like the pipe. In the pipe it looks like this, that's the pipe. That means inside the pipe there is the hollow part. So the cisternae look like this, but it is a branch, it's not a straight. Like it is, the cisternae look like this. So that means a pipe like structure or flattened like structure of ER is called as the cisternae, and another structure is called as the tubule. So what is the tubule? The tubule means, the as the name suggests, it is a tube like structure. It looks like this. Here also it is a tube like structure. So they are called as the tubule. So that means inside the cisternae and the tubule, inside the hollow part, that means inside like the pipe, inside there is a hollow part in the cisternae as well as in the tubules. So in the ER part, there are the classified again the ER as luminal, and another is the extraluminal. So what is the luminal, what is the extraluminal? So luminal means inside. So extraluminal means cytosolic. So what is the inside? For example, some molecule is present here. So that means it is present inside the ER. So that means we can say that this molecule is present luminally. That means this molecule is present inside the endoplasmic reticulum. So what is the extraluminal? That means cytosolic. Simply it is the outside of ER. So let's suppose the molecule is present here. That means it is present outside the ER. So now we can say that the molecule is present extraluminally. That means the molecule is present in the cytosol or in the outside of the ER. So this is the luminal and the extraluminal part of the ER. So now come to the major difference between the, or major structure of ER. The ER can be classified, or ER can be divided into two parts. One is called as the rare and another is called as the sere. The rare is called as the rough endoplasmic reticulum, and sere is called as the smooth endoplasmic reticulum. So what is the rough endoplasmic reticulum, or why it is called as the rough, why it is called as the smooth? Look at the structure here, from here to here. So in this structure, a blue color structure is like this, they are present, and these are called as the ribosome. So that means in the rough endoplasmic reticulum, the ribosomes are present. So because of the presence of the ribosome, it is rough. While in the smooth endoplasmic reticulum, look at this structure. So in this structure, there is no ribosome. So that means that's why it is called as the smooth endoplasmic reticulum, or it is called as the sere. So this structure is called as the rough endoplasmic reticulum. because of the presence of the ribosome on the membrane of the rough endoplasmic reticulum. So if you look the endoplasmic structure, it emerges from the nucleus. This is the nucleus, this is the ER, and this is the Golgi. So look at the structure. The ER emerges from the nucleus part, and that too nucleus has, we have already discussed that nucleus is the double membrane bound organelle. That means the nucleus has two layers. One is called as, this is called as the outer layer, and this is called as the inner layer. So that means the ER emerges from the inside or the inner layer of the nucleus. That means, let's say this is the first nucleus, and from this the ER emerges like this. Let's say this is the nucleus. The ER emerges from the inner membrane of the nucleus. So this is called as the rough endoplasmic reticulum because of the presence of the ribosome, and the smooth because they don't have the ribosome. So this is the main difference between the rough and the smooth endoplasmic reticulum. So why the ribosomes are present? Actually, in the ribosome, in the rough endoplasmic reticulum, there is a protein is present that is called as the ribofourin. Ribofourin, that is a protein present on the rough endoplasmic reticulum, and that helps in the attachment of the ribosome. That means it's simply, you can say that it's a like glue-like structure. That means this protein which helps the attachment of the ribosome in the rough endoplasmic reticulum. So now come to what is the function of the rough endoplasmic reticulum. That means what does it do? So the rough endoplasmic reticulum, it helps in the protein synthesis. Protein synthesis. That means in the rough endoplasmic reticulum, it helps in the protein synthesis. Please look at this term. It helps in the protein synthesis. In the NEET exam, there are the two types of the question it will be asked, that if the question is which organelle helps in the protein synthesis, the answer will be the ER. But the second question is, what is the site of protein synthesis? So that means, what is the site of protein synthesis? That means on where the protein synthesis is occurred. So the answer will be not endoplasmic reticulum. The answer will be the cytoplasm. So that means in the rough endoplasmic reticulum, it helps in the protein synthesis. It synthesizes the protein, but the protein synthesizes exactly in the cytoplasm like this structure. So once the protein is synthesized in the cytosol, now it enters inside the rough endoplasmic reticulum. But first it synthesizes in the cytoplasm. Please remember this. If in the NEET exam it will be asked that what is the site of protein synthesis, the answer will be cytoplasm. But of course the rough endoplasmic reticulum helps in the protein synthesis. That means rough endoplasmic reticulum with ribosome, it helps in the protein synthesis, but protein synthesis occur exactly in the cytoplasm, and after the formation then it enters inside the endoplasmic reticulum. So this is the main function of the rough endoplasmic reticulum. And which type of cell contains the rough endoplasmic reticulum? Mainly liver, pancreas and fibroblast. So that means the liver of course it contains many enzymes, many proteins, many lipids. So because of this, means liver is responsible for the formation of many enzymes or many proteins. So that's why liver contains high amount of the endoplasmic reticulum. Along that, pancreas is also site of the production of the many proteins and the lipids. And the fibroblast means it's simply a connective tissue. It synthesizes like the collagen. The collagen is synthesized by the fibroblast. So the fibroblast also contains the endoplasmic reticulum. So now come to again first rough endoplasmic reticulum. It synthesizes the protein at the outside. Let's say this is the protein and it is protein. This protein is synthesized at the outside. Now after that it enters inside the endoplasmic reticulum. So that means the first it is the extraluminal, and after the production it becomes the luminal or inside the endoplasmic reticulum, and after that it forms a vesicle like structure. Vesicle means simply a structure which is surrounded by a membrane, and inside the vesicle the protein is transported. And now this protein, this is a protein, so now this protein enters into the golgi. This is the main function of the rough endoplasmic reticulum. That means it is helps in the protein synthesis, and after protein synthesis it transports to the golgi by forming a vesicle like structure. Okay, this is the rough endoplasmic reticulum. These all are the rough endoplasmic reticulum because of the presence of the ribosome on its membrane. So another is called as the smooth endoplasmic reticulum. So why it is smooth? Of course it is, they don't have the ribosome and they have only the tubule like structure. Look at the structure. Here it is called as the smooth endoplasmic reticulum. Only the tube like structure is present. That means only the tubules are present in the smooth endoplasmic reticulum. So what is the main function of the smooth endoplasmic reticulum? So it is the site of lipid synthesis, lipid as well as steroid, lipid and steroid synthesis. So that means the rough endoplasmic reticulum, it helps in the protein synthesis, and the smooth endoplasmic reticulum, this helps in the lipid synthesis or lipid and the steroid. So now which cells contain the smooth endoplasmic reticulum? The cells like liver, a fat cell, and adrenal cortical cell. So adrenal cortical cell you can simply call as adrenal cortex. So what is the adrenal cortex? You will know about the hormone part, that in the adrenal there are the two parts, the two layers. One is called the inner and it is called as the medulla, and the outer is called as the cortex. In the cortex part, it synthesizes three hormones. One is called as the glucocorticoid, mineral corticoid, and another is the androgen. So these all are the steroids. So that means since the smooth endoplasmic reticulum, it is responsible for the lipid and the steroid synthesis. That means the liver, of course, it also contains, it also helps in the, it is means responsible for the production of protein and the lipid also. So like that fat cell, of course, it synthesizes the lipid, and the adrenal cortical cell, that means the glucocorticoid, mineral corticoid, androgen, they all are synthesized by the cortex. So that means the liver, fat cell and the adrenal cortex cell, they contain the smooth endoplasmic reticulum. Look at the structure. Here also, let's say this is the lipid is present. Here also, like the rough endoplasmic reticulum, it also forms a vesicle like structure. And inside it, a lipid, let's say it is the lipid. Inside it, the lipid is present, and now the lipid is transported again into the Golgi. So what is the function of rough endoplasmic reticulum and the smooth endoplasmic reticulum? Rough endoplasmic reticulum function is the production of the rough protein, and after that it forms the vesicle and transports to the Golgi. Smooth endoplasmic reticulum, it synthesizes the lipid and the steroid, and then it forms the vesicle like structure and it's transported to the Golgi again. So another main function of the smooth endoplasmic reticulum is it helps in the detoxification. It is a NEET question. Many times it will be asked that. So which organelle helps in the detoxification? The answer will be endoplasmic reticulum. But if in the option that is called as the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi, lysosome, then answer will be smooth endoplasmic reticulum. The smooth endoplasmic reticulum has one more extra function, is it helps in the detoxification. And detoxification is occurs in the which organelle? It is the liver. Maximum detoxification occurs in the liver. That's why liver also contains high level of smooth endoplasmic reticulum. Okay. So this is all about the rough endoplasmic reticulum. So now come to the Golgi body. It is called as the Golgi body, or it is called as the Golgi apparatus. So, or it is called as the Golgi complex. You can say whatever you can say, that Golgi body or Golgi apparatus or Golgi complex. So this is the structure of the Golgi. So the Golgi, it was discovered by Camillo Golgi in 1898. So the first electron microscopic structure of the end of Golgi body is look like this. That means it is a densely colored structure that is found in the electron microscope. Look at the structure, and this is the Golgi body, and it was seen in the electron microscope for the first time, and it was densely, means a very black color or very darkly stained, the color of the Golgi body was seen. So that's why it is called as the densely colored structure that was present inside the cell is called as the Golgi apparatus. So the Golgi apparatus, they found in the all the cell except mature RBC, sieve tube and sperm of bryophyte and pteridophyte. Okay, so that means mature RBC, sieve tube and the sperm of bryophyte and the pteridophyte, they don't have the endoplasmic reticulum. So in the animal cell 3 to 7 complex are present. 3 to 7 means this is the 1, this is 2, this is 3, this is 4. That means 3 to 7 complex structure of the Golgi are present, but it is found very large in size in case of the animal, but in the plant the number is 10 to 20, but the size is very small. So that means the animal, the number is huge, but number is very low, but the size is very huge. But in the plant the number is very huge, but the size is very small. And that's why in the plant the Golgi body is called as the dictyosome. Dictyosome. So that means what is the dictyosome? It is simply the Golgi body that is present inside the plant is called as the dictyosome. So come to the structure of Golgi body. This is the structure of the Golgi body. The structure contains three types of the structure are present. One is called as the cisternae, one is called as the tubule, and another is called as the vesicle. So what is the cisternae, what is the tubule, what is the vesicle? The cisternae means it's like this flat like structure, they are called as the cisternae. The flat long disc like shape, flat disc like shape is called as the cisternae, and its the size is 0.5 to 1 micrometer. What is the micrometer? Simply the 10 to the power minus 6 meter is equal to 1 micrometer. So that means 1 meter by 10 to the power 6 is 1 micrometer. So this is the diameter of the cistern. So that is another is called the tubule. What is the tubule? And at the periphery there are many structure looks like this, and this is called as the tubule. And another is called as the vesicle. Vesicle, that means this is a vesicle. So that means a structure in which it is surrounded by a layer and it contains something, that is called as the vesicle. So there are the three types of the structure. One is the cisternae, a long like structure. The tubules, a very small, that is present at the periphery, is called as the tubule. And the vesicle, that means from the Golgi the vesicle arises and it contains some food particle. We will know something after this Golgi body study. We will know about the what is the vesicle. So now come to the cistern. It is very important. Cisternae means, look at the structure. This is the nucleus, this is the ER, and this is the Golgi. The Golgi part, it has a two part. One is called as the convex and another is called as the concave. Means let's say this is the nucleus, this is the ER, and the Golgi is present, looks like this. Look at the structure. That means one part is called as the convex and one part is called as the concave. You know what is the convex, what is the concave. The pointed end is called as the convex. So that means one part is here, this part is called as the convex and this part is called as the concave. So the convex part is also called as the cis face, while the concave part is also called as the trans face. That means here it is called as the cis, here it is the trans. That means here this part is called as the cis part, and here this all the large structure they are called as the trans part, or it is called as the Now, a forming phase, and it is called as the mature phase. Why it is called as the forming, why it is called as the mature? Means if in the child the size it looks like this, but after the adult, of course the size increases. So that means it is the forming phase, it is the mature phase. Here also look like this structure. That means it is a forming phase. That means it is just formed here, but it is the already matured one. So that's why the trans part is called as the mature phase. And always remember that this C phase is facing towards nucleus. Look at the structure. This phase always looking towards the nucleus. That means it is nearly present at the nucleus in comparison to the trans phase. So that means this phase is very far in comparison to this C phase. So this is the C phase, this is the trans phase. The C phase is the convex, the trans is the concave, and the C is forming phase, and the trans is mature phase, and the C is towards the nucleus, and the trans is away from nucleus. Okay, so they are interconnected. That means the Cs and the trans, they are interconnected here. So this is the main structure of the Golgi body. So now come to what is the function of the Golgi body. So that means in the ER, from the ER, the proteins are transferred by forming a vesicle. Here in the smooth endoplasmic reticulum, the lipids. The lipid, here it is the protein. So they are synthesized and transported to the Golgi. So in the Golgi, the C phase accepts the protein and the lipid. So always important, the C phase accept the protein and the lipid and the steroid. Okay, so what is the function of the lipid, function of the Golgi? That means after accepting, what will he do? So look, let's say this is the protein. Now the protein comes inside the Golgi. Now the Golgi forms the modification of this protein. That means it helps in the modification of protein and lipid. That means let's say this protein, this enters into like this, here inside lipid, here it also it enters like this. So after this it modifies this protein and the lipid. So what is the modification? The Golgi body adds a carbohydrate into him. So the process is called as the glycosylation. So what is the glycosylation? That means the addition of the carbohydrate to any molecule. So here in the protein and the lipid, the main function of the Golgi is it adds a structure here also, it is called as the carbohydrate. That means it modifies the protein and the lipid by adding the carbohydrate, and the process is called as the glycosylation. The second is called as the most important protein sorting. The cell sorting. Sorting means, let's suppose this is a cell, this is a nucleus, this is ER, and this is the Golgi. So that means in the ER, the protein and in the smooth endoplasmic reticulum, the lipid are present, lipid are transported into the Golgi, and after that the glycosylation occurs, and after that it decides this protein or the lipid should go to where. That means, let's say there are the many organelles, for example the mitochondria and lysosome, and many organelles are present. Let's suppose this protein is for the mitochondria. That means the Golgi decides whether it should go to the mitochondria or not. If it decides it goes to mitochondria, then this structure, that means after the glycosylation, the structure, it also forms the vesicle like this structure. Here, this is the vesicle. The vesicle like structure, now it enters into the mitochondria. Let's suppose it decides this protein or the lipid should go to the membrane, then it forms again the vesicle, and this vesicle fuses to the plasma membrane. Looks like this. This is the protein or the lipid that is needed for the plasma membrane, so it fuses like this. Let's suppose it decides this protein or the lipid should go to the lysosome, so again it forms the vesicle. So now this vesicle, it fuses to the lysosome, and now lysosome should get its protein or the lipid. So this is called as the protein sorting. That means it decides which protein or the lipid should go to the which organelles. So that's why the Golgi body is also called as the traffic police of the cell. So like that means in the traffic police, like that, the traffic police decides which side of the people should go to where. So like that, the Golgi also decides which molecule or the protein should go to where. So this is the main function of the Golgi, and along with that it helps in the formation of cell wall by dictyosome. So that means in the plant the Golgi is called as the dictyosome, and the Golgi of the plant or the dictyosome helps in the formation of the cell wall in the plant cell. And another is it helps in the acrosome formation in sperm. So what is the acrosome formation? That means during the fertilization, let's say this is the ova, this is the sperm. So it is some reaction is required for the fertilization, and this reaction is called as the acrosome formation. So the Golgi in the sperm it also helps in the acrosome formation that is needed for the fertilization. So this is the main function of the Golgi. That means accepts the protein and the lipid, and after accepting it modifies by adding the carbohydrate, or the process is called as the glycosylation. Then the cell sorting, that means it decides which protein or the lipid should go to where. The next one is it helps in the formation of the cell wall by the dictyosome, and another is it helps in the acrosome formation in the sperm. Okay, so this is the all about the Golgi apparatus. So now come to the another structure that is called as the lysosome. Okay, the lysosome, it was discovered by Christian D. Dubé in 1955. So it is, of course, it is a single membrane bound organelle. That means it is bound by a single layer, and it is formed by the process of the packaging in the Golgi apparatus. For example, look at the structure. This is the ER. So this is the Golgi, and this is the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, it synthesizes the lipid and it transports to the lipid, it transports to the Golgi. And here also the smooth rough endoplasmic reticulum, it synthesizes the protein and transports into the Golgi. And after that, the Golgi apparatus sorts, that means the protein and the lipid, it should be decided by the Golgi apparatus. So now come to the lysosome. So like the packaging, the lysosome, this is the lysosome. So that means the lysosome is also formed by the process of the packaging of the Golgi body. So what is the function of the lysosome? That means here also this lysosome is also formed by this Golgi body. So what is the lysosome? It simply, let's say this is the lysosome. It contains many hydrolytic enzymes. Hydrolytic enzyme. So what is the hydrolytic enzyme? That means it is a digestive enzyme or simply acidic enzyme. So that's why, what is the pH of lysosome? Always it is the very acidic, because the hydrolytic enzymes are present, or the digestive enzyme. For example, protease. Protease, that means it digests the protein. Carbohydrate, that means it helps in the digestion of the carbohydrate. So nuclease, that means it helps in the digestion of the nucleic acid or the DNA and RNA. For example, the lipase. So lipase means it helps in the degradation of lipid. So that means all these hydrolytic enzymes or all these digestive enzymes are present inside the lysosome. So first, next come to the what main important is the polymorphism. Polymorphism of lysosome. So what is the polymorphism? That means more than one form of lysosome. That means the lysosome have different type of the lysosome are present. So that is called the polymorphism. So it is also a NEET question. That means which organelle shows the polymorphism? The answer will be lysosome. So the lysosome are the three types of the lysosome are present. One is called as the primary lysosome, another is called as the secondary lysosome, and another is called as the autophagosome. The secondary, it is also called as the heterophagosome. The primary is simply called as the primary. So what is the primary lysosome? That means in this primary lysosome, here also the hydrolytic enzymes are present, but the hydrolytic enzymes are present in the inactive condition. Inactive means here also the enzymes are present, but it is not active. For example, in this structure also, let's say any protein comes to this, it cannot digest because the protease is present, but the protease is present in the inactive condition. So that means in the primary lysosome, the proteins are present, but it is present in the inactive condition. But in the secondary or the heterophagosome, the protein is present and that too in the active condition. So that means here all the molecule, that means protein, carbohydrate, lipid, nucleic acid, phosphate, all these things, they are digested by this secondary lysosome. So that's why the secondary lysosome, the main function is the digestion of the food. So what is the digestion of the food? Simply let's say in the stomach. Let's say in the stomach. In the stomach cell, if I cross-check it, there are many different types of the cells are present. So if in the exam it will be asked that which type of the cell or in the cell, let's say this is the one cell of the stomach cell. So in the cell of the stomach, which organelle digest the in the stomach the proteins are digested. That means in the cell of the stomach, which organelle digest the protein? The answer will be the lysosome. So that means the lysosome of the stomach digest the protein. So that means in the liver also, in the small intestine also. So that means they all contains the lysosome. That means inside in these liver, small intestine and stomach, that means all the alimentary canal, that cell contains the nucleus, the lysosome more vigorously, because this lysosome helps in the digestion of the food. Another important is that lysosome which also helps in the digestion of foreign particle. What is foreign particle? That is simply called as the antigen. For example, let's say this is a bacteria or the virus or the bacteria. So what is the foreign particle? That means anything which is not about the cell or not about our body. That means outside body material. Let's say this is the bacteria, it enters into it. So now this bacteria, the lysosome digests the bacteria because the lysosome contains the acidic enzyme, the hydrolytic enzyme. That means this hydrolytic enzyme can digest anything. So it also digests the bacteria also. So that is the main function of the lysosome. One is the digestion of the food. Along with that, it helps in the digestion of the foreign material, the foreign particles such as bacteria, virus, pollen. What is the antigen? Different types of the antigen, they can be digested in the lysosome. And another is the autophagosome. Autophagosome means, let's suppose this is the nucleus, this is the ER, and this is the Golgi. So this is the lysosome. This is the, let's say, mitochondria. So if the mitochondria is damaged, so now that means it is now not in use in the cell. So this mitochondria, it fuses into it and it enters into the lysosome, and lysosome digests its own cell because it is not now the use of the cell. So this autophagosome, it digests its own organelle which are damaged or dead, of course. That means it is not use of this. So that means the autophagosome is digestion of its own organelles. So that's why the lysosome is also called as the suicidal bag of cell. It is very, very important. It is many times asked in the NEET exam that which cell organelle is suicidal bag of the cell, and the answer will be the lysosome, and that too autophagosome one. Okay. So this is the all about the lysosome structure of the cell. So now come to the vacuole. So what is the vacuole? Vacuole is also a membrane bound organelles, and that too single membrane bound organelle. Okay. So this is the, let's say this is the plant cell and this is the vacuole, the blue color hole, the part, it is called as the vacuole. And the plasma membrane of the vacuole is called as the tonoplast. So what is the tonoplast? The tonoplast is simply the plasma membrane of vacuole. So what is the role of this tonoplast? Okay, before going to that, what is the role of the vacuole? So the vacuole contains water, sap and the excretory product. So what is the sap? Sap means simply any liquid part that is present in the plant is called as the sap. So water, sap and the excretory product, and the vacuole contains this type of material, which is, interestingly, which is not useful for the cell. Not useful for cell. Okay. So vacuole contains this water, sap, excretory product that is not useful for the cell, and the vacuole are very, very large. That means, look at the structure. This is the mitochondria, looks like very teeny. This is the nucleus, it is also very teeny. But look at the vacuole, the structure. That means in the plant 90% of total volume is occupied by the vacuole in the plant cell, of course. That means the vacuole is so much large that it enters or it occupies 90% of total volume of the plant cell. But in the animal cell, normally in the animal cell, it can be found or it can be absent. So that means if it is found, it is present in the very less in number and very, very small, very, very small. That means in the plant cell, the vacuole looks like this. In the animal cell, in the animal cell, the vacuole looks like this. But in the plant cell, this is a very huge, this vacuole. So what is the main function of the vacuole? The vacuole main function is this is the tonoplast, and through the tonoplast, the ions of the solute are transported into it through active transport. So what is the active transport? In the previous video we have studied about the active transport. That means it uses the energy and the solute, it transfers from low concentration to high concentration. So that means here the ion concentration, let's say this is the ion, for example sodium, potassium, the ion concentration is very low, and inside the vacuole the ion concentration is very high, and this solute or these ions are transported into this by this membrane, and this membrane is called as the tonoplast. So through the membrane of the tonoplast, the ions are transferred by the active transport, that means by the use of the energy, that means from the lower concentration to the higher concentration. So in the NEET exam it will be asked that what is the concentration of the ion in the inside the vacuole? Always it is very, very high, because it always transports the ions inside it by the active transport. Okay. So now the vacuoles are mainly two types. Vacuoles are mainly two types. One is called as the food vacuole and another is called as the contractile vacuole. So what is the food vacuole? Food vacuole means simply it digests the food like the lysosome, but less than 1%. That means maximum digestion of the food occurs in the lysosome, and very small amount of the food is digested in the vacuole. And another is that it found in protozoan, protist and lower animals. Well, what is the function of the contractile vacuole? Look at the amoeba. Let's say this is the amoeba. So in the amoeba, here the contractile vacuole. That means it regulates the osmoregulation. What is the osmoregulation? It is simply the osmotic pressure. What is the osmotic pressure? That means it decides, this pressure means the pressure between the solute and the solvent. Simply, you can say that in the animal body, if the water level is very high in the body, so after that we transfer the water by the peeing, so that the water amount should be labeled. So if the water amount is very low inside our body, so we drink water, so that the water amount should be same. So here also same function like the contractile vacuole. That means inside the amoeba, let's say suppose the water level is very high, so it discards the water to the outside. So if the water level is very low, then it don't allow to discard of the water to the outside. So this is the main function of the contractile vacuole, which is found in the protozoans, for example the amoeba. So this is the all about the vacuoles, and this is the all about the endomembrane system, which includes endoplasmic reticulum, golgi, lysosome and vacuoles. Okay, so if you like the video, then please like and subscribe. It will take maximum 2 seconds. Thank you.